we are looking at ecological sampling and specifically how it is being measured. We've had a look at different kinds of things that we can measure, but now we are really looking at sampling, specific sampling in the field. So taking a sample is a way to see a small selection of something larger to get an idea of what the entire thing is like. I can sample a meal, I can do a 3-month trial of Stan, I can survey a group of people in the school about their opinions on the tuck shop, whatever it is, right? Depending on how I take this survey, it can give me an idea of the general opinions of the entire school population. Now, ecological sampling is a way to get a small snapshot of an ecosystem, which can best represent the complexity and diversity of a wider ecosystem. It's meant to be representative. To sample an ecosystem, a small area within the ecosystem is surveyed and we collect abiotic and biotic data about the area. It might just be, temps, salinity, pH, dissolved oxygen, light intensity, whatever it is. Or it might mean that we are counting organisms, calculating their density, drawing profiles, whatever it is. Choosing the survey technique that are relevant to the area is really, really vital. So you don't study, say, grassland the same way you would study a rainforest, okay? The life is pretty drastically different there. The density, the light intensity, all of that stuff. You have to make the correct choice as to what technique you are using. From a bigger picture perspective, choosing a wider strategy to make sure the sample is a good representation of the ecosystem is also really important. So it's obviously impractical to consider counting every single organism in every single habitat, so the sample has to be representative. I keep coming back to that word. We sample things so that we can monitor, manage, conserve an ecosystem, and this involves monitoring populations of organisms, plant, animal, classifying ecosystems, whatever it is. We need to be able to do sampling for many, many reasons, and so there's so many different whys. There's no one why. We need to collect quantitative and qualitative data so that the sampling can provide a full picture of everything we're trying to look at too. And we can look at things change over time to monitor when we need to. If we go back to the opinions around the tuck shop, ideally we would want to survey the entire school, but that's impractical. But if I only surveyed the Year 12s, this wouldn't be a good idea either. So it's important that the sample fairly represents the demographic mix of all students. There's that word again, representative, right? The same goes for ecosystems. Where there's a variety of habitats sort of within an ecosystem, it's important to sample in a way that accurately represents the entire thing. We take a selection of samples from all across the ecosystem to make necessary assumptions that this is showing us what the whole thing looks like. So this is a different way of doing things as opposed to this. If I only sampled the back end of this population, I am not going to get a truly representative sample, so that's not helpful. There are a few ways to design a sampling strategy which ensures that the sample taken is fair, accurate, and has minimal bias. And we'll discuss a few of these strategies as well as the techniques. So methods and techniques, my apologies. These are the specific kind of nitty-gritty things that you might have even seen in junior science, and these are designing your experiment in a bit of a different way. So we'll talk about the sampling techniques first, so that when we discuss the overall strategies, you'll have a more of an understanding of what these might look like. So quadrats, first of all, it is a frame of known size. It can be rectangle, it can be square, it can be circle, whatever it is, it's usually square, which marks out a particular area that's being sampled. And they're placed in an ecosystem. The species within them are identified and recorded. It might be, not a literal square because you can't put that over the tree, whatever it is. Could be made of plastic, wire, could just be 4 pegs put in the ground to make a big square, whatever it is. They can be placed randomly in a habitat, can be chosen using a random number generator and a grid reference, whatever it is. They're used to collect counts of organisms. You can measure abundance, you can measure presence or absence, you can measure population density, percentage cover, percentage frequency, so many things just from this one square. So they're pretty good for plants and animals with really low or slow mobility, not, a bilby running straight through it. Now a transect line is a line might be an actual physical piece of fishing line, I don't know, placed through a community which acts as a boundary on which the sample is taking place. So the sampling might be taken exactly on the line, underneath the line, to the right of the line, whatever. You could use a tape measure, it could be a marked line, it could be with different pegs, whatever it is. It can show changes in abundance and distribution along a gradient. So you might go from high up altitude to low, you might move from the shore out to the ocean, whatever it is. You're looking at how the plant life, the abiotic factors, whatever, changes perhaps along that line. And you can sample systematically along that environmental gradient. So if you're sampling on a line transect, you walk the entire length of the line, you record the type, the position of every species on the line. It might be very time consuming, or you might just place a quadrat alongside the line as well. There's a little bit of leeway given on either side of the transect line, or you could just do the the quadrats on right, left, whatever it is. You can systematically put these lines down, or you can randomly do them so that they might end up overlapping, but this is a truly random way of sampling. So a belt-transect, uses a pair of parallel lines, and it works in the same kind of way, but maybe every single thing within those two lines is sampled, and they can be placed in rows, regular intervals in that belt as well, so that everything inside is recorded and everything outside is not recorded. Now, a point transect collects data at regular intervals along that transect line, and this is kind of like a belt transect, but in a much smaller range. So they're mainly used to calculate percentage cover or density, and you can see this is still a systematic way of doing it, and it's using both quadrat and transect. Sampling on a transect line can lead to an accurate scale profile diagram being drawn up, like a side view or an elevation, and the shape of the land can also be included to see elevation and other geographical features. In places like forests and with layers of vegetation, you can see them all in that profile diagram, like the canopy and the understory in a rainforest. Now, we've spoken about mark and recapture in reference to estimating population size. So there are other techniques mentioned. They focus really heavily on vegetation, but mobile animals are always a bit of a challenge as they move in and out of your sampling area, and they might just be completely scared off by you entirely if you're there with a big quadrat. So using the Lincoln Index, we can capture these animals, mark them, maybe tag them, we release them back, and then we come back at a later date and take a second sampling. And the proportion of marked or tagged animals in that second capture is used to estimate the entire population size. Now, of all the techniques just mentioned, they're all smaller tools to use within a wider sampling design. So when we're talking sampling methods, it's like, how are we organizing this? How are we designing this experiment? Because that's what's happening here. So if I talked about the students at the tuck shop again, they're going to rate it from 1 to 5. That's a technique. But how I go about doing that is the method, the design. If I choose students just in the class that I'm speaking to, that would be one method. I could talk to every 5th person I walk past, that's another one. I could ask 50 students from each grade. The type of sampling method used in ecology, they're kind of similar, right? And any other kind of statistical or population sampling would have to think this through as well. This is the big picture overall approach stuff. So random sampling can be done if time is really limited or you're in a really large uniform area. It's not really so good for clumped distribution. It can involve random selection of data. So the entire population, each possible sample is probably equally likely to occur. And at a basic level, the quadrat can just be kind of thrown down randomly, throw it around and, pick it up. But, human elements always come into play. I'm going to throw it towards that tree. Oh, it's bounced off. So we tend not to do it like that. It's also really really challenging to randomly sample things because you are seeing what you're doing. So it's best to split the area up into a large grid, use a random number generator to decide which quadrats are going to be sampled. You might mark the corner of the quadrat to encompass, larger species, whatever it is. And logistical challenges, sure, but it's trying to avoid bias each time. Now, systematic sampling although the name suggests otherwise, it's actually a type of random sampling, and the same sampling can occur. It might be quadrat, it might be a transect line, whatever it is, but it's happening in a more structured way. So you might be using a fixed periodic interval. It's agreed upon in advance and adhered to. It's like, yeah, every third line here we're using. It might mean that every 5 meters along your transect, you are putting down a quadrat, or every second quadrat in the larger grid is sampled. So if a species shows a really clumped, non-uniform distribution, using this is probably a more appropriate way to make your data more meaningful. And choosing between random and systematic will heavily depend on what ecosystem you're in, and probably be relative to what you're trying to measure. Now, opportunistic sampling is convenient sampling. It's exactly that, right? It's using convenient, easily accessible sites to survey. It's a non-random technique, so it can exclude an entire chunk of the area, and it's not going to be representative of the entire ecosystem. You usually do it to get preliminary data really quickly. So if I just went, oh, I need to know about the tuck shop. Hey, year 12s, can I ask you about this, please? Because they're there in front of me. And then we decided, oh, there is actually a problem with what people think of the tuck shop. Let's go ask the entire school population. Now, stratified sampling is where a proportionate number of observations are taken from each part of the population. So you have to divide an ecosystem into zones, and it appears, different, take samples from each zone, and then strategically sample an ecosystem in a way that's more representative. So if you've got an area that, oh, this is slightly bigger, I'm going to do more of them here. This is slightly smaller, I'm going to do fewer zones in this area. Now, some really specific syllabus stuff here. Part of choosing a sampling technique and method is making sure you are choosing it for the right reasons, considering the most appropriate sampling method and sampling technique, right? Is this the most appropriate way to sample this environment? Each one is going to come with its challenges. It's going to come with different, what species are in there? Are they mobile? Are they easy to count? Are they easy to identify? So making sure the technique and the method is the most appropriate for that area is going to be really important. Each technique is going to have its pros and cons, and it's worth considering them as we learn about them so that we can design, say, student experiments. And this is a real bigger concept that we'll look at in class as well. And added on to this are the ideas around validity and reliability of that sampling technique and method. So these are concepts you'll have to have an excellent handle on for your field design, your field work design for your student experiment, but it's also good to consider around basically every piece of data provided. Has the right technique or method been used given the environment that's been surveyed, and is this going to negatively impact the validity or reliability of different sampling techniques? Oh, I have to go out and do this survey again because I lost my dog ate my homework or whatever. Am I going to get similar results because of how I have approached this and the method I have used? So definitely these are ideas that are going to keep coming up in student experiment designs for fieldwork in particular. Lots going on here, lots to consider, only a little bit of time to have to do it, but it's going to need to be applied to a number of different data from fieldwork tables, I suppose.